It's wonderful to be here. I'm always delighted to talk to the friends of ISB as well as my colleagues. Today, I'm going to talk to you about the future of cancer immunotherapy as we're building it here at ISB and with many colleagues. One of the great pleasures that I have as a scientist and a professor is to work with all sorts of wonderful colleagues, but also a host of really terrific students. And I'll highlight the students' contributions as I get through this talk. And also I want to acknowledge on the right, I've listed the various foundations and government agencies and individuals that have been so supportive of this research. Without that kind of support, it certainly would never happen. Okay, so immunotherapy, it's a type of cancer therapy that probably, it goes back, I think, to the 90s, but it wasn't really until about 2010 or 2015 that it really began to take over cancer with a basically therapy with a storm. And here you can kind of see why. On the left, I show a PET scan of a patient who had highly metastatic melanoma, and you can see these just a god-awful number of lesions here. These are lesions that are metabolically active, and so they show up in a PET scan, but these are just metastatic lesions from a melanoma tumor. And this patient was placed upon what's called checkpoint inhibitors, a type of cancer immunotherapy that doesn't actually attack the cancer cells. Instead, it releases the immune system to attack the cancer cells. And then over a period of 1 year, you can see that these tumors are gradually destroyed by the immune system. And we know it's the immune system doing this because if you actually look at the tumor in one of these patients that's responding so well, and it doesn't always work this well. In fact, one of the big challenges is to get it to work this well. But you see these T cells, this purple world of T cells that just coming in and attacking the tumor compartment. It's a truly remarkable, basically, piece of science, working its way into the clinic and bringing great benefit to patients. And so what we would like to do, and I use the we as a community, is to try to figure out how to bring similar benefits to patients that have all kinds of solid tumors. By and large, tumors of the blood system, leukemias, there have been great strides against leukemias with things like CAR T cells and bone marrow transplants, cord blood transplants, etc. Solid tumors are still very, very challenging. Melanoma has been an early success story. There's certain lung cancers that are success stories, but most solid tumors are still very challenging. And so in a solid tumor, what you're trying to do is to get T cells, these gray entities here, to recognize and attack a cancer cell. And this molecular structure I show here on the right, this is actually a couple of protein complexes that are brought together by this peptide antigen. And the peptide antigen is just a little fragment of a protein that is associated uniquely with the cancer. And so there's, billions and billions of T cells in your body. They all are different, but some of those T cells, their antigen recognition region will detect this peptide as it's supported on the cell surface by what's called an MHC molecule. So the heart of all of cancer immunotherapy, by and large, is how do you get this to happen in a patient's tumor? And one of the beauties of cancer immunotherapy is if you do this correctly, these T cells are only going to attack the tumor, nothing else, because that little antigen, that little mess of balls here, that's just a small peptide, is what makes that tumor unique. And so if you can identify an antigen and a T-cell receptor and an MHC molecule that can lead to a therapy, then the process for turning that into a cancer therapy, pretty high tech actually, but it's become more and more standard over the past decade. You take T cells out of a cancer patient, you'll knock out the T cell receptor that T cell once had, and you'll knock in a new one that specifically recognizes that antigen presented by that MHC molecule. You'll build those T cells up, put them back into the patient, and just watch the tumor melt away. That doesn't always happen, but that's the goal of this type of therapy. Now, the way this therapy has been developed has, for completely innocent reasons, really, has had favor for, I guess, you could say, patients of European descent, and it really neglects other ethnic groups. And the reason why is actually scientific, not so much anything evil, and it's because this MHC molecule here is something that is unique to patients. Each of us have 6 of these MHC molecules. They all present different types of antigens, different variations of peptides that could be from a virus or from a tumor or something like that. 3 of those MHC molecules we get from mom, 3 we get from dad. If you put a group of 10 people in a room together, you would find that a couple of those MHC molecules would be common amongst a few of those people, okay? But typically, these MHC molecules are common for just one ethnic group. So as an example, here's these MHC molecules are labeled by these various numbers, A21, A11, A30, et cetera. So a very common one for European descent individuals is this A21. And here I show it for, it's got a fair amount of abundance in the European population, but almost none in the Asian, a little bit more in the Southeast Asian, and a little bit more in African descent communities. A1101 is sort of the opposite. It's very high in Asian communities and Southeast Asian, but not really represented otherwise. But because A21 is so broadly represented in Europe and the US, that's really where the focus of research has happened, because that's where you get statistics from patients and stuff like that. But as a consequence, the types of treatments that have been developed really neglect non-European ancestry patients. And that's something that we're working to try to rectify. So this diversity of MHC molecules and that diversity that is, distributed across different ethnic groups really suggests that if you want to have a immunotherapy that treats all solid tumors, at the very minimum, you'll need to have T-cell receptors that recognize tumor antigens in patients of European ancestry, Southeast Asian ancestry, African ancestry, Pacific Islander ancestry, et cetera, suggesting that you actually need a whole library of T-cell receptors. No one of these is going to be effective at treating a population of patients. So that goes counter to how this type of therapy is developed now. And I'm really focusing on these T-cell immunotherapies because when they have worked in solid tumors, they've worked spectacularly well. And I think most folks are thinking that as we move into the future, these types of therapies are probably going to be the most effective for eradicating solid tumors. But in any case, to do this, one first identifies a protein that is only in cancer cells. You identify a fragment of that protein that's presented by the MHC molecule. You'll make some sort of fish hook that's designed to capture T cells that recognize that peptide fragment and MHC molecule, and then you'll use it to pull T cells out. You'll identify a T cell that recognizes that antigen. You'll clone the T cell. He'll ask, does it actually kill tumor cells? Does it have autoimmune consequences if you were going to give it to a patient, et cetera? This is a one-off process, and it literally takes years just to get one T cell. So, what we have basically is this thing on the left, which shows how books were obviously reproduced many, many years ago. And what we need is a really high throughput technology for developing this library of T cell receptors against cancer. So this is this one slide is fairly busy, but I just want to sort of illustrate a pathway that my lab has taken over the past, just 15 years now. So about 15 years ago, we had a technology that we reported that allowed us to look for not just one antigen and one MHC molecule in one patient, but we could actually look at something like 12 antigens. About 9 years later, we came up with an approach that allowed us to look for 50 antigens, just one MHC molecule, one patient, but 50 antigens, so that's better. Last year, we reported an approach that allowed us to now look at up to 3 MHC molecules, still just one patient, but something like 1,000 antigens. So that's a 3,000-fold, or 3,000 divided by 12-fold improvement. And then this year, we have an approach that allows us to now have a 1,000,000-fold improvement. And on top of that, for each cell that we get about a 1,000,000-fold increase in the information content per cell. And with a technology like this, it simply allows you to ask questions differently. And so that's basically a technology that we're taking forth to develop a T-cell receptor-driven immunotherapies for initially for cancers that are driven by human papillomavirus, but other cancers as well. So the reason why we're focusing on human papillomaviruses with driven cancers with this technology, and that includes a lot of cancers, it's head and neck cancers, cervical cancers, anal cancer, several others, is that the HPV virus itself actually drives the cancer. And so there's 2 proteins in this virus that are going to be in every single cancer cell. And those proteins will get chopped up. They'll be presented as antigens by the MHC molecules on the cancer cell surface. That's something that looks foreign. It's not part of your normal self-biology. And so T cells can potentially recognize those viable fragments that are presented by the MHC molecules, attack the cancers, and kill it. And in fact, the proof of principle of this whole concept has been illustrated by Christian Henrichs, who came out of the NIH and is now at Rutgers. And in fact, we're working with Christian on this project. So one experiment that this technology that we've developed in my lab allows us to do is to look at, for lots of HLAs, lots of antigens, lots of patients, to just begin developing this T-cell receptor library that one can draw upon to treat virtually any patient that walks in the clinic. And so this was an initial experiment that we did just at the end of last year, largely by Rongyu Zhang and Rachel Ng in my group, but also Jingyi Ji was involved in this, and several people are involved in this going forward. We worked with Connie Trimble from Johns Hopkins, who has a biobank of patients that have precancerous HPV-positive lesions. And in that analysis, we looked for lots and lots of antigens. So this is obviously way more than one. And then out of that, we pulled out T-cell receptors that we're in the process of validating right now. And for each one of these different colors here, that's a different MHC molecule. So already this type of experiment is allowing us to begin identifying T-cell receptors that can be used to treat a broad ethnic diversity of patients. And in fact, we are moving this into the clinic. This is a project that ISB is leading. I'm the scientific lead on this project, but it involves now 3 different medical centers: the Swedish Cancer Institute here in Seattle with the City of Hope in Duarte, California, and UCLA, and my longtime collaborator, Tony Rebus, down at UCLA. And we've just now advanced our first T cells into an engineering runs that will hopefully let us file with an IND with the FDA by a few months away, and maybe begin treating patients by this fall. So it turns out that as we begin to apply these types of technologies that I've just referred to understanding how the immune system responds to cancers, we're finding that we don't just need a T-cell receptor library that allows us to treat all patients, regardless of their ethnic background, but we actually want a T-cell receptor library that allows us to engineer what we think of as a balanced anti-tumor immune response into the patient. So in other words, we don't want to just engineer the T cells to kill the tumor. We want to engineer the T cells to remember the tumor, so that if it comes back, you have immunity against it. It keeps it from coming back. You want to engineer against therapy resistance, and there's certain kinds of therapy resistance that I think are very common that we can readily engineer against. And so one thing that we've turned to is the sort of modern computational biology method, something ISB is very well known for, and asked, can we develop computational approaches that will help accelerate this whole library construction? And so there's two questions we want to ask of this computational tool set. And the first question is the following. When a T cell binds to an antigen MHC that's presented by a cancer cell, so the T cell has seen the cancer cell, the T cell can do lots of things. It can kill that cell, and that's called a cytotoxic response. It may not kill the cell, but on the other hand, it might remember the cell, and if it sees it again, it may signal for more T cells to come in and kill the cell. So the functions of immunological memory and the functions of immunological killing are not necessarily done by the same T cell. In fact, they're clearly not. And so can we predict, if we know what that T cell receptor is, we know what the antigen is, can we predict what the T cell will do? And the person who's led this, in fact, the computational, this one and the next one, is the student of mine, Daniel Chen. This is one of the examples of really having the pleasure of working with bright people. Daniel started at UW as an undergraduate student when he was 14 years old and began working in my lab shortly after that. He's now a Marshall Scholar at Cambridge University. He's just 19, but he's got a very impressive publication track record from the work he's done with us. So here's the question. So we have this MHC antigen at the top. We have all these different T cell receptors below, and it turns out you can have lots and lots of different T cell receptors that will recognize that MHC antigen. But after they recognize it, what are they going to do? Are they going to be cytotoxic, or are they going to have memory? So I'm going to cut quickly to the chase on this and not really walk through the algorithm that Daniel developed, but I can tell you that this has been a very deep literature of trying to understand what is it that influences the outcome, the properties of a T cell once it sees that tumor antigen. And there's lots of things that can be important. What cell actually presented the antigen to the T cell? What's the local biological environment in the tumor? What are the different MHC molecules that are present? Obviously, as we've talked about, those are going to vary by patient ethnicity, but even within an ethnic group, they're going to vary from patient to patient to patient. And finally, what is the genetic code of those T cell receptor genes that recognize that antigen? And we're the ISB. We measure lots and lots of things in lots and lots of patients, very complete data sets. And so we actually can address all of these factors. And what we found, and we've just recently published this, and so reports about I think it came out maybe just a couple weeks ago, is that the only thing that really matters for at least certain classes of viral antigens, which are very relevant to HPV-driven cancers, is the T-cell receptor gene. So what I plotted here is if something mattered, I guess, zero amount, it would be here at a zero. If it matters more than expected, it's going to be a high number. And if it matters less than expected, it's going to be a low number. And the T-cell receptor, the blue thing here, is the only thing that really matters. That's really important because what that means is that when we go to our library and pick out T-cell receptors for cancer therapy, we actually know what those T-cells will do when we put them into a patient. So it allows us to think about designing a balanced immune response, one that both remembers the tumor and is cytotoxic against the tumor. The second question we asked, and this is, I think we'll walk through this a little bit more, is a more challenging question. So we have about 1 trillion T cells in our body. And to first approximation, every T cell has a different T cell receptor. We also have about an equal number of possible peptide antigens that can be presented to those T cells. So if we had a huge database of antigens and a huge database of T cell receptors, can we predict which T cells will recognize which antigens? So this is the question. Lots and lots of antigens, lots and lots of T cell receptors. And answering this question is really a grand challenge of T cell biology. And so I'm going to divert a little bit and talk about the innovative algorithm that Daniel developed, but give you a couple of analogies to give you some insight into what that algorithm represents. So over the past 3 or 4 years, there have been artificial intelligence tools, the same things that go into something like a ChatGPT model, but that have been applied to understanding crystal structures that have been collected for the past, 70 years or so. And out of all that database of protein crystal structures, being able to now calculate a protein crystal structure without actually, this is as close to an experimental measurement as you could possibly imagine. So, for example, if you crystallize one of these proteins here, you would know where all the atoms are to, say, a root mean square deviation of 1 angstrom. Very, very tiny amount, about the length of a carbon-hydrogen bond. Computationally, you can generate that same level of accuracy. And that's because we have all these known protein structures. And so we can predict other things that are similar to these structures. And that kind of approach, I think, has been one of the more transformative things that's happened in biology over the past decade, for sure. But with poorly understood biology, this approach actually generates bad guesses. And the question we're trying to answer, which T cell receptors go with which antigens, is still poorly understood biology. So let me give you a simple analogy on how this algorithm was developed. So if you think about a large language model, so this is a term that gets used a lot in artificial intelligence. It's basically the types of models from which something like ChatGPT learns how to answer questions and things like that. So let's develop a large language model that's designed to predict how balls will bounce around. Drop a ball, you want to know what's going to happen. You can train on the motion of cannonballs. And if you predict on the motion of cannonballs, you're going to get a pretty good prediction. So this plot here shows the value of that prediction. It says you get the answer right about 86% of the time. If your answer was along the diagonal, it would be half the time, and that means it's a coin toss. You basically have no predictive power. If it's below here, that means your prediction is actually worse than a coin toss. And if it was all the way up here at the top, it would be perfect. Okay. So let's take that same large language model that we predict the motion of cannonballs and now use it to predict the motion of ping pong balls. It's going to be awful. We're going to get no accuracy at all. So instead, let's develop a large language model that uses at its heart some of the fundamental physics that was developed by Isaac Newton. Force equals mass times acceleration, work equals force times displacement. Every action has an equal and opposite reaction. That's obviously a ball bouncing when you drop it. And if you take those equations and you train them on cannonballs and you predict cannonballs, you're going to do phenomenally well. If you train them on cannonballs and you predict ping pong balls, you're still going to do pretty well. I haven't thrown in friction here into these equations of motion, so we're not going to do perfectly, but we're going to do well. Okay, so can we take a large language model like what's used in ChatGPT, reduce it in size so that we're only capturing sort of a fundamental rule set? So here's the analogy. With ChatGPT-like large language models, you would take the entire database of T-cell receptor antigen MHCs that's known, maybe 100,000 are known, and you would look at all the information content that you have associated with that and try to develop a predictive algorithm. That's millions to billions of variables. It's a very, very computationally intense algorithm. If you take a fundamental rule set, what we do instead is we take the amino acids of those peptides and of the T-cell receptors that are in contact, and we put in, from many years, we actually understand the hydrophobicity, meaning how attracted or repelled to water are these amino acids, the biochemical features, the structural sort of steric issues, the charge, etc. So we get a much smaller set of variables, and it's drawing from the fundamental biochemical, biophysical characteristics of these amino acids. But for both of these language models, we use the same neural net algorithms to make the predictions. With this model that Daniel developed, we actually guess right on a large data set about 90% of the time. Pretty fantastic. If we compare how this works against the other models, and here I've shown for 3 common what are called immunogenic antigens that come from Epstein-Barr virus, cytomegalovirus. So Epstein-Barr virus, many of you all probably familiar with this. It's not only associated with mononucleosis, but also with MS. A cytomegalovirus, a common virus that many of us carry. And influenza, everybody knows influenza. Our predictions here are the blue, and these other models are predictions are the other colors that are basically just literature models that use these types of ChatGPT large language models. So clearly, our predictive power is very high, but on top of that, the amount of time it takes for us to do these predictions is something like 1,000 times faster than competing models, because it's a small data set that we're using scientific smarts rather than just data to do this calculation. And furthermore, because our calculation involves a relatively small number of variables compared to these big ones, you know, one of the mysteries one hears about these AI algorithms is no one really knows how they work. Well, we actually know how this works. And out of it, we can extract design principles. And so what that means is that we think in the not too distant future, we will actually be able to design a T-cell receptor for our library for cancer treatment just by knowing what antigen we want to bind to, and not having to actually do measurements on patients. We think we can do it completely in silico, as they say. So I'll conclude there. The technology for identifying T cells or recognize specific tumor antigens, it's improved, dramatically, many millionfold in terms of information content. And ISB and my group, we've been leading that tech development. Computational advances are equally rapid. And I think because we're now beginning to extract design rules, we may be able to design the T-cell receptors we want from scratch, which basically then puts no limit on the size of the T-cell receptor library that you can build. And then those T-cell receptors are just on the shelf, and you use them when you need them for therapy. So these off-the-shelf TCRs can be used to make TCR-engineered T-cell cancer immunotherapies more effective. They can expand the benefits of cancer immunotherapy to all patients, et cetera. And we are working very hard to rapidly move these concepts into clinical trials to treat patients, hopefully with the first patient being treated by the initial steps of this type of concept this year. And finally, AI won't yet replace scientists, at least not for a little while. And with that, I thank you for all your attention, and I will stop sharing, and I am delighted to be able to answer any questions people might have.
Thank you, Jim. That was fantastic. There is some discussion in that Q&A, some questions. I've compiled a few. One question was, you mentioned the clinical trial on HPV cancers. Can you describe that a little bit more about what's going on there?
Sure. So we're working with City of Hope, UCLA, and Swedish Cancer Institute to do this. We have identified an initial TCR to go into that trial, plus we're developing the pipeline of TCRs so that as we learn how to use this type of therapy, we will be able to expand and use the repertoire of TCRs in ways that I described in my talk. There's a lot involved in this. So we've done a lot of preclinical validation of this, and we're beginning to do that on the other TCRs. We've actually converted a clean room facility in here into making the genetic material that's used to engineer the T cells, and then we've contracted with a organization on the East Coast. So we'll take blood from the patient. That organization will take our plasmids, engineer those patients' T cells to attack the tumor. That takes about 2 weeks, and send it back to us. So it's a pretty rapid process, and we'll initially be treating patients both here and at City of Hope, and as we expand to multiple TCRs, we'll be expanding to include UCLA as well.
Wow. So there's a question about accessibility. Clearly, what you described to me was impressive, that you can now design these therapies for different ethnic groups. You can discover these TCRs that have never been characterized before, and the question of affordability comes up in terms of how much does it cost, and when do you think it will be affordable for a person going to a community clinic to see an oncologist?
Yeah. So as you can imagine, the early versions of this therapy are a lot more expensive because you have to use, an excess of caution. We know these therapies tend to be pretty well tolerated by patients, but you have to prove that in many, many patients before but I think ultimately this is an outpatient treatment. Further, you have the issue. So right now, the FDA makes us file a separate IND, meaning a separate filing for a new drug for every single TCR that we identify and want to advance to the clinic. That is a huge burden. I think over time, and on top of that, there's no standards. Many places across the country, companies, academics, what have you, are trying to do these kind of therapies. The Hutch has some, Eric Tran did amazing work with prostate cancer. Every approach is one-off. And so there's been no standardized approaches for not just identifying what T cells are important, the whole validation manufacturing approach. So something that we're trying to set up here is to really streamline that. Much of that can be done with robotics. So, and then you have the issues of engineering the patient's T cells. Well, initially that was a very challenging thing to do, but now, we have CRISPR methods for knocking in the T cell receptor and knocking out the endogenous T cell receptor in one step. That now is looking like it can be a reasonably modestly priced approach. So I would imagine that in a few years, this will be about the price of targeted inhibitors, something like that. Now, it doesn't mean you can always trust pharma companies or whoever's going to really take this to a large scale. But experience in this in biotech tells you that if you have something that really does scale to lots of patients, just the scale of that is going to put competition and bring the price down. So I'm optimistic that this can be competitive with other treatments. And if we do it right, it's curative. So it's not like you're sitting there going from one therapy to another, to another, to another. Hopefully you're going to be done. Now that'll take some time, obviously. But I think, we and others have a line of sight on how to make that happen.
Right. Now, when we think about cancers, we always learn about two dimensions. One is interpatient heterogeneity, and the other is intratumoral heterogeneity. What you described essentially addresses, to a large extent, the interpatient heterogeneity. But I have two questions that have come up. One is, do you think there will be eventually some rules that you'll learn that might allow you to stratify patients in a way that you can find some commonalities, so you can generalize some of these therapies to subgroups of patients, then instead of having one therapy for one patient?
Well, I think it's going to be, so the way I look at it, so we already know what some of the rules are. We just don't have the tools to take advantage of those rules. So I'm going on the premise that we'll have lots and lots of T cell receptors, and it's not going to cost you any more to use 2 or 3 than it is going to cost to use 1, because they're all just on the shelf. They're known quantities. So for example, many patients, I talked about the fact that you have these 6 HLA molecules, 3 from mom, 3 from dad. As tumors advance, oftentimes they will actually quit producing some of these HLA molecules. You'll lose the ones from mom, but if that happens, the ones you got from dad are very protected, or your whole immune system will go in and kill those cells. So let's take T cell receptors that are specific to cancer antigens on something from mom and one from something from dad. You avoid that's a I think you would do that on every patient, even if you don't have that type of heterogeneity, even if that patient hasn't lost that, had that type of resistance mechanism activated yet. But why not? It's not gonna make it that big of a difference. I think also you've got to be very careful that with these HPV cancers, we know the HPV proteins are in every single cancer cell. And so that's a challenge. So you have to know when you're going after targets on these cancer cells that those targets are really in every cancer cell. And that's where a lot of patient-to-patient heterogeneity comes in for a single type of cancer. So you go for what are called oncogenes, the driver genes that really dominate a tumor, or things like that. And that's probably where, for some tumors, you may run into roadblocks with a technology like this. So far, there aren't any clear killers, and especially if we get design rules for how to design a T cell receptor, in silico, I think some of those are circumventable, but it's hard.
Yeah, no, but yeah. So along those lines, I mean, do you have any predictions as to which cancers would be the next best targets after the HPV cancers?
I think I don't know, but I can give you a little bit of guidance. So over the past, the cancer, the ability to look into what makes a cancer tick has really changed dramatically over the past handful of years. And a lot of that's because, now we can take a piece of tissue, and on every single cell in that tissue, we can look at its genetic material. We can look at those genes that are being expressed. We can look at the state of the chromatin. We can look at the proteins. And we can do that on, obviously lots and lots of patients. Those types of studies are going to give us the deepest insight into how and when we direct immunotherapies for tumor eradication. So I would almost say that tumors that get really deeply studied in that way are likely the next candidates. There's not something intrinsic about one cancer or another that seems like it's worse. I think some cancers like glioblastoma, brain cancers, as you know, there's other barriers that are going to make that very challenging. You've got the blood-brain barrier. But the one thing immunotherapy does do that most therapies don't is it actually travels to the site of the tumor. If you can get the drug into the brain, or you can get it into the blood or whatever, you hit distant metastases as easily as you hit the primary tumor. So, yeah, I mean, I think name your cancer, you're going to come up with a whole bunch of reasons why it's really hard to do. But I think you'll also come up with a whole bunch of strategies for how one could solve that cancer.
Yeah, and in brain cancers, I mean, I heard of the Omaha Reservoir where you can directly deposit some of these therapies to the site. And there's also, you can do microwave radiation-based slight breaching of the blood-brain barrier to let cells go through. So yeah, as you say, there would be challenges, but I think on a case-by-case basis, there might be solutions as well.
Yeah, I mean, I think this is why we got excited about immunotherapy many years ago, is that for almost any cancer, one can come up with a hypothesis for how to treat it. It might not be a good hypothesis, but one can come up with, and you can't say that about any other approach.
Yeah. There's more questions than I can cover, so I'm going to shift a little bit here. So there's a question from Terry Bergeson, our friend. A great question, and I think you'll have a great answer because I know some of the work we're doing here. Is there a way that science teachers can bring these new learning into their teaching in the near future?
Oh, absolutely. So I think, we at ISB, we have developed a number of these modules. I think Nitin has really led this, but there's also been sort of efforts from the whole. We have several education teams here that develop education modules that go out into the community, even around the world. I think this issue of molecular recognition, which is actually a really cool issue. These molecular structures are beautiful. I mean, and thinking about how they interact with each other, the new computational tool sets and ways you can bring to visualize that are actually not expensive. Some of these, like the algorithms that I talked about today, to do a very sophisticated calculation takes 1 minute on a pretty good platform, but on a PC, it's going to take maybe 10 minutes. And they're just going to get better. And the visualizations, which are available, through, relatively simple technologies as well, I think are very powerful. I mean, these are kind of things that one could even explore using iPhones or iPads or things like this. And I think it's actually a really good idea for us to think about how would we take some of these concepts, some of these technologies that we're learning, and turn them into such education models. ISB has a long history of doing that, and I guess we've been so on this freeway going 90 miles an hour. We haven't thought about how to carve this out into educational opportunities, but there are many.
Well, there are some. I mean, so we've been working with the Office of Superintendent for public instruction, the OSPI office in the Seattle public school systems, to develop systems medicine curriculum. And one of the modules that essentially focuses on personalized precision medicine and cancer immunotherapy. So there might be an opportunity to carve out some of the lessons based on some of the learnings from your work. So
Oh,
I certainly
absolutely.
see a direct path there. Yeah, yeah.
And this kind of thing actually touches on not just cancer, but infectious disease, autoimmune disease, everything. So, and we do have education models that are actually very comprehensive on health, wellness, and even navigating the healthcare system. And so this is a place where that kind of thing could fit in.
Yeah, so we're at 12:54. Maybe we'll do two more questions. Um, so you addressed this question in a more scientific perspective, but this may be a more administrative, logistical, and policy perspective. Is what are the challenges of getting this type of research into the healthcare system?
So to launch and really carry through a project like this in a scientific laboratory like ours, it's really expensive. And so certainly cost is one. And right now, biotech is not seeing a good moment in the commercial space. And so that's a major thing. The second is that you find, I think this is where ISB really excels, is that, I walked you through high-end computation, and I walked you through biophysics and biochemistry. I walked you through patient care and bioengineering and fundamental immunology. Um, those kind of concepts are generally siloed in most places. And I think the ISB is really good in that we don't do that kind of thing. So there's a lot of artificial barriers to this kind of thing as well. But to get CAR T cells and checkpoint inhibitors have shown that if you have success, you can get it into a healthcare system. Now, it doesn't mean you get it uniformly into the healthcare system as well. And we have even curative immuno-oncology treatments that are readily available are not used everywhere, and that has to do with sometimes it's rural urban division, sometimes it's underserved and well-served communities. Sometimes it's just physician knowledge on how to deal with side effects. We have projects at ISB that are addressing these various bottlenecks, and I'd be delighted to talk to someone in more detail about this. I think that's an area where one can make great progress.
Great. So maybe the last question. So the question is, aspirationally, what do you hope cancer therapy looks like in 10 years or 20 years?
Well, I think in 10 years, I think we will have a much deeper understanding of identifying patients who are likely at risk of cancer relative to those who aren't. Right now, if you've got BRCA1, BRCA2 mutation, things like this, you can get targeted early on for more monitoring. But our knowledge of human genetics and genetic risk is growing extremely rapidly. And, for like $50, you can get enough of your genome sequence to understand that risk. That's like parking in some places. And so I think that's one thing, because in any time you can detect a disease early, whatever you're going to try to do to that disease, you're going to be more effective at stopping it. Second, I think we'll have a much deeper understanding of the immune system. And, we may look back on these engineered T cells as sledgehammers, and we may find that, in fact, for early disease, we will have ways to activate the immune system that are much more subtle. And I would hope that would happen. I think we're going to have to go through some sledgehammers first. And these are like little tiny tapping hammers compared to chemotherapy. But I think we're going to learn a lot. Right now, our knowledge of human immunology is probably doubling every year. And I think it's The kind of things I talked about, the different T cells for memory, for cytotoxicity, for signaling, et cetera. We didn't even know you could do that like 3 years ago. But now we know, and we almost have the design rules for them. So I think the world's going to look really, really different. I think it's going to be about identifying patients who are at risk very early. If you're not at risk, don't worry, don't go to the doctor. If you're at risk, you're going to have a motivation to go and have simple tests done. And then if changes are happening, I think we'll have immunological interventions that will be subtle. They don't need to be hammers at that stage. So I think it'll be some form of what I talked about, but subtler and gentler and more anticipatory.
Great. Jim, this was really fantastic. And thanks for taking the time to share your research on the future of immunotherapies. There's the questions, there are so many, I couldn't cover them. So I apologize to those who took the time to sit through the presentation and share their thoughts. And I hope we have a record of it because I think you'll enjoy going through those questions. There's some really good ones. So from the questions, I could tell that a lot of you, hopefully all of you, enjoyed Jim's talk as much as I did, or maybe more. And I hope that gives you a flavor of the kind of leading edge research that's going on at ISB. And as I said at the beginning, the reason we can do some of the research at the leading edge is because of generous contributions from people like you. So if you are encouraged today to go and make a gift, I certainly welcome that. And I would direct you to ispscience.org/donate. And do keep an eye on your email inboxes because we have this series on a quarterly basis. So the next event will be announced very soon. So I welcome you to join us again. And thank you for joining us today, and have a great, wonderful rest of your day.